My name is Tom Peavy, and I'm a faculty member in the Biological Sciences Department here at Sacramento State University. And I want to welcome you all to our first Topics in Regenerative Medicine seminar series, the first one of our semester. This seminar series is co-sponsored by the Center for STEM Excellence, which STEM in this case stands for Science, Technology, Engineering, and Mathematics. And it's a center that promotes the advancement of sciences on our campus. And it's also, this seminar series is funded from California Institute for Regenerative Medicine grant, and they, also known as CIRM. And speaking of which, Alan Trounson had come and visit us, who's the president of CIRM, last spring. And so maybe you caught that seminar, but if you didn't, we also, the Center for STEM Excellence has a website that you can visit all the past lecture series, including this one tonight. And CIRM is also supporting our stem cell research program, which is a collaboration between our biology department here at Sacramento State University and UC Davis stem cell program. And that's supported by a Bridges to Stem Cell Research grant. Students in this program undergo 5 semesters, which in essence is about 20 months of the program. They get their education here at Sac State, and then they perform the research in stem cell laboratories at UC Davis. Those kind of research labs are such cutting-edge research disease teams like Huntington's disease, HIV, diabetes, and cancer, which happens to be the topic of today's talk. So if you're interested in our program, you can come see me afterwards. I'm the academic coordinator, and I also have flyers at the back of the room or where you entered into this room. So having said that, it's my great pleasure to introduce our speaker, Dr. William Murphy, today from the UC Davis Medical School. And Dr. Murphy is very active in our program, also in our Bridges to Stem Cell Research, and he houses 2 of our students currently in his lab. And his research program is more centered on the immune system, and in particular, immunotherapies targeting the eradication of cancer stem cells, which are resistant to conventional radiation and chemotherapeutic treatments. And Dr. Murphy first started off his career by receiving a Bachelor's of Science degree from Rutgers University in biology, followed by his PhD in immunology from the University of Texas Southwestern Medical Center. And then Dr. Murphy went on to his postdoctoral studies at the National Cancer Institute at Frederick, Maryland, and ended up becoming a director for their basic research program. Then he, in 2002, joined the faculty at the University of Nevada Medical School in a department of microbiology and immunology, and became the chair of that department in 2007. And fortunately for us, he's now accepted a faculty position has been at the Department of Dermatology and Internal Medicine, and he's serving as the acting chair currently in that department. Dr. Murphy has had a very productive career and has published in more than 200 peer-reviewed publications over the last 30 years. And today he's going to share with us his research on the use of natural killer cells for an immunotherapy to attack cancer stem cells to increase the success rate for eradicating cancer. Please join me in inviting Dr. Murphy to give us a talk.
Okay, thank you very much. That was a very nice introduction, and I'm really pleasure to be here and meet such excellent students. I've been very happy with the CIRM Bridges program, and the Sac State students have been really outstanding. And what I'm going to talk about, which is a little bit different, is where stem cells are bad. So most of the time with CIRM, you're thinking about good stem cells, and I'm going to talk about when stem cells go awry, but this might actually help put things in context, because it's kind of amazing when you think about the complexity of cancer. Ever since Nixon declared the war on cancer in the 70s, we've been trying really, for the longest time, trying to increase our cure rates. And it's been very difficult, and I'm going to show some data with some statistics. But for the first time with immunotherapy, there's an excitement. In the past year, it's exploded. And we're finally using the C word with the C word. We're using cure with cancer, which we never used to do when I was at the National Cancer Institute, mainly because now we're finding out when we use the immune system, we actually can eradicate or at least control the disseminated disease, which is the crux of what the cancer problem is. So I'm going to talk about some of our work looking at cancer stem cells, which is kind of a new paradigm shift on how we approach cancer, because for the longest times we were treating cancer as one disease. I find it kind of amazing when you think of the fact that ACS, the American Cancer Society, predicts that in the next couple of years, the cancer incidence in the United States will be 1 in 2. That means there's a 50% chance that you're going to know somebody directly or yourself will have cancer. And yet, for the most part, when you think about it, most people have a high school biology education when they're having to confront this disease and figuring out the treatments. And it's kind of scary in that sense, because in the era of personalized medicine, molecular medicine, the families are at the forefront trying to figure out, what do I do? How do I treat it? What are my options? And now we're understanding truly now that there are some potential options there, but this is a really complicated disease. So when we're talking about cancer in the United States, what a lot of people don't realize is that there's a difference between incidence and death, or mortality. And by far, with incidence, skin cancer has the largest prevalence, but for the most part, with basal cell and squamous cell, you're not dealing with a metastatic disease situation. A lot of people will get it, particularly as you age and in the high sunlight areas, but for the most part, you're looking at, as far as the mortality rate, it's melanoma, which is a relatively rarer cancer. A lot of when we're talking about cancer, we're talking, and at cancer centers, we're talking about what we call the big five, in as much as the most prevalent. And these are the solid cancers, the epithelial cancers, the lung cancer, colorectal, breast, pancreatic, prostate. These are the ones that have the highest mortality rates and also have a significant incidence, as you can see here. And that's what most people don't realize too, is every cancer is so different. It's hundreds and hundreds of different diseases. I remember when I was at the National Cancer Institute, and for a certain kind of leukemia, a drug, a molecular targeting agent, was shown to be efficacious. And the cancer center director said something very interesting, because the drug had no side effects and really worked in remission. He said, this is going to hurt us as far as with cancer, because now the public's going to expect it to work for every cancer. And that's not been the case, particularly with a lot of the solid organ cancers. This is looking at death rates in the United States as far as with cancer, and you can see all the way from the 1930s, when it comes to the major cancers like breast cancer and colorectal, we are seeing a slight increase as far as our survival rates, but for the most part, when you consider the amount of resources, the amount of energy, amount of money, amount of time spent on this disease, you're really not seeing a drop-off in the mortality. And in fact, a lot of this is being actually attributed not so much for the treatment, but we're picking it up better. We're doing better screening. And by doing better screening, we're actually being able to eradicate. Cancer is a disease where the more you have or the longer you have it, the worse it is. It's that simple. So if you do an early screening, early prevention, and this is why we're seeing the drop-off in lung cancer, simply because of the cessation of smoking. But this tells you, with the advent of something like smoking, the amount of years it takes to actually impact a population, much less than when we start to see the cessation of smoking, particularly in males, not as much in females, we're starting to see a lower mortality. But this is telling us, too, that it takes a long time to figure out when we're making a difference with regard to cancer treatment. Now, if I asked everybody in this room to define cancer, I'm probably going to get a lot of different answers. And one of the things that I found when I teach this class, I teach a graduate class in cancer immunology at the medical school, is that everybody has this sort of idea of cancer, but as far as certain jargon, you hear the words neoplastic, transformation, tumor, malignancy. All of those are correct, but all of those sometimes are rather loosely applied, and really in the definition of cancer, when we talk about it, it's hard not to use those jargon in describing it to a layperson. And so one thing I have my students do right away is define cancer. And when we think of cancer, and by the way, this is the reason that we call it cancer, is when you look at this is the symbol of the National Cancer Institute. When you look at a tumor macroscopically, it becomes invasive, it spreads out, literally looking like a crab when you start looking at it as far as within the tissues. But cancer is a genetic disease. And when I say genetic, I don't mean that you're inheriting it. Yes, you may inherit susceptibility genes as far as repair and be prone to it, but it actually comprises of permanent genetic mutations that occur over long periods of time. And a lot of people don't realize that long periods of time, because cancer evolves, it develops. And there's a big difference between cancers in adults versus kids with regard to the level of complexity. Commonly, to be truly malignant, it has to be invasive. may not be metastatic, but it has to penetrate as far as in the break the basement membrane and be invasive to start spreading. Otherwise, you hear the words benign versus malignant. And depending on where the tumor is, if it's in the brain, for example, it can be benign, but it can eventually grow enough where it still can be fatal. But for the most part, invasive and metastasis is the hallmark of cancer, and it's metastasis that ultimately causes a lot of the mortality. The hard part is when you think about cancers, how do you know when you have it other than screening? It's usually symptoms like pain, bleeding, and other things. And unfortunately, by that time, usually metastasis has already occurred, even at a micrometastatic level. This is a very nice study done at Johns Hopkins over a period of time, and what they found, at least with colon cancer, you're dealing with something that can occur within, it takes 20 to 40 years to develop. Now think about that. And what that means for me is that I might have something right now that has been developing for 20, 30 years, and I may not even know about it. And that's also very attractive when you're thinking about the immune surveillance theory, is that the immune system may be keeping it at bay, but as we get older, maybe our immune system starts decaying, and therefore the cancer emerges. But what we're seeing is cancer is a result of multiple mutations that occur over periods of time. And it's these mutations that make the cancer so formidable, because these mutations help it evade the immune system, help it actually become more mutation prone, and also help it spread. And so these are the things when you talk to a person, they say, oh, I've had cancer. Some people think it's that quick, that they just found out and it's been growing just for like a week or two when they get a diagnosis, when in reality it can be long, long periods of time. If you're an immunologist, and the whole concept of immunology is how we fight foreign pathogens, this becomes very tricky. And cancer, literally to an immunologist, may be the ultimate enemy because it arises from normal cells. And the immune system, as a rule, has been educated not to attack or induce autoimmunity to your own body's cells. And the cancer then develops, and we don't know when we can actually use the immune system to actually see that and use it to our advantage, because it's the immune system that can maybe seek out and find out where those metastatic lesions are. Otherwise, when we're talking about typical cancer treatments, you're talking about cytoreductive conditioning, which is chemotherapy, radiation therapy, and surgery, the conventional treatments. But that, unfortunately, that's not usually targeted or specific. And so at some point, the cancer may relapse, and this is where we need the immune system to mop up the rest. Now, when we draw cancer pictures, like in my class or on my little whiteboard, when we think about how we're going to cure cancer, we draw pretty much all the cells look the same, and it's all like real wiggly looking and mutated. But when you're looking at cancer in the real world, you're looking at successive changes in the cell architecture, and ultimately what the diagnostic test used for cancer is histology. They do a biopsy. And the biopsy will help them determine as far as what kind of cancer it is, how aggressive it is, staging it and grading it. And then also, even when you have, in this case, liver metastasis that originally arose from colon cancer, they're going to be able to then figure out where the primary tumor is. So when you think about the daunting kind of problem that a physician has is, where is the cancer? And using imaging, you can pick up some of it, but you're limited because you're only seeing visually what we call macroscopic. It's very difficult to actually see what we call micrometastasis, and that can reappear over long periods of time. This is considered, how do you create a cancer cell? You need to understand your enemy if you're going to fight it. And again, a lot of people may think of one tumor cell that lives forever, and that's not the case. If you ever look at a primary tumor, there's a lot of dead cells in it. The cancer cells do die, but they just keep proliferating. Normally, with normal cells, there's a periods of, they can only proliferate so many times, and then they undergo senescence. Cancer has avoided that. And that just keeps going and going and going. In fact, our tumor lines that we use in our research, some of these tumor lines have been from a patient that was 40, 50 years ago. The HeLa cells are the classic example of that. And they just keep growing and growing. That's what we mean by immortal. Immortal is not that this one cell keeps living, but it just keeps giving rise to more and more of the cells, and it becomes autonomous. That means ultimately when you're thinking about cancer, you're thinking about something that is not behaving normally and under normal growth control. It's that autonomous that keeps going on its own tune that makes it so difficult to treat. If I yelled something like fire right now, I didn't mean that in that sense, but if everybody just stampeded out That's what cancer is. Every cancer cell has got its own tune and is looking out for its own individual survival, whereas in the body, normally with our cells, they cooperate. And if they're not doing the right job, or if they're damaged, they undergo apoptosis or death. Cancer evades all that, and then has a huge advantage over the normal cells. Now, this is a classic paper by Bob Weinberg in Cell, in which he describes this is what cancer, if you want to create cancer, these are all the different things that have to happen genetically for it to occur. If this does not happen, you do not get cancer. All right? And it seems kind of simple, but when I was at the National Cancer Institute, this is just oncogenes. So when I was at the NCI, oncogenes were the big thing. Oncogenes, 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 that's the cure for cancer. But then we found that the body also learns to shut things off, and cancer has to evade that. Then we found out that cancer actually has to become immortal. That means it can keep going no matter what. But then angiogenesis, remember, I don't know if those of you remember, maybe about 10, 15 years ago, angiogenesis was it. The cure for cancer, was on Time magazine, was going to be blocking angiogenesis. Well, it didn't really work out that way. It's not as simple as that. It does play a role. Angiogenesis is the forming of blood vessels. Cancer needs air, needs oxygen, needs nutrients. You block that, you starve it. But cancer also has all these other advantages versus, say, normal cells, including resisting cell death. So that means that cancer is normally able to resist a lot of our means to attack it by the immune system and other mechanisms. And all these things together give you cancer, including the invasion of metastasis. You need that to get the spread. Unless you're having cancer in a vital organ, it's the spread that will ultimately kill you. However, now this is being modified. It's not as simple as even that. And these are what we call the emerging hallmarks. That means, what Bob Weinberg, that issue of Cell 10 years ago was the most highly cited article in Cell's history, which, when you realize the impact of that journal, says a lot. But now he's updated it, and he says, you know what? Cancer evades immune cell destruction. And if you have a healthy immune system, or if the cancer has something expressing on it that the immune system can see, you don't have the cancer. It's eradicated. And just like a virus, the cancer has developed ways to evade, suppress, and avoid the immune system. Inflammation, no pun intended, is really hot. It's a hot area right now because what we're finding is inflammation actually induces carcinogenesis, induces tumor promotion, and induces tumor spread. Believe it or not, aspirin is making a comeback as far as in breast cancer and other areas, not only for the prevention, but also for the treatment of cancer. So where inflammation looks to be really key. Cancer also, this is the old Warburg hypothesis, it doesn't need oxygen as much. It lives in a hypoxic area for the most part, and the cellular energetics is off, where it goes more by anaerobic rather than aerobic. And this is a big one, genomic instability. When I was at the NCI, we were also at the NIH working on HIV. And the cure for HIV, not the cure, but as far as what controlled it, was triple drug therapy. They found that the virus, even though it's a simple retrovirus, could only mutate at a certain rate. And if you applied 3 highly HART or antiretrovirals that work differently, but at the same time, you could control the virus. Now that's something simple like HIV. Cancer has the entire human genome at its disposal. But not only that, cancer by its very nature is very unstable genetically. So that means when you give chemotherapy, usually you're going to get a relapse, but when you get the relapse, the tumor is now resistant to that therapy. It's mutated or selected out variants that can now then take over, and therefore gives you even more limited options. But even as an immunologist, this genomic instability of cancer is the biggest hurdle. Because a cancer cell, if it doesn't need it, it loses it. And if it loses it, that could be your target. And then ultimately you'll get relapse. But these are now the emerging hallmarks, and our laboratory has been interested in looking at the immune system. But wait, there's more. The other thing that we're finding out is that cancer is not as simple as we thought. All the cancer cells are not the same kind of cell, even though they're all genetically different as far as mutagenicity. What they're finding is even when they start looking at primary tumors versus metastatic tumors, it can give rise to different kinds of mutations. And that makes this a very difficult cell to attack. The other thing that makes it very difficult is what we're finding, and this is the new paradigm, cancer stem cells. You may have heard that word a lot. Now, it's very different. I'm going to go into what a cancer stem cell is. But it is this kind of heterogeneity that is making it even more difficult to target a tumor with a one-size-fits-all drug or approach. And this is where we're starting to see more and more personalized or individualized medicine, where everybody's cancer is unique because everybody genetically is unique, and it's not one-size-fits-all. Cancer stem cells. What we're finding is that their tumor cells are heterogeneous, but how do we study it? Normally, what we do is in vitro and in vivo assays. In other words, you can't just tell the patients, roll up your sleeve. I can give, as an immunologist, I can give everybody a vaccine here to flu, and everybody's going to react differently to it. Similarly, your cancers are all going to be different, and also how you react to different drugs are going to be different. So what we use are in vitro assays, which involves tumor cells that are grown in culture and are from original human tumors. Now, that's an artifact, okay? We grow these cells under optimal growth conditions over long periods of time. The other thing we do is in vivo assays, where we can actually study cancer in mice, and then also start to look at the immune effects. But one thing that people have noticed is that when we start to look at these models, we get a better idea on really what kind of tumor cells it takes to grow either long-term, in vitro, or in vivo, and we're finding that there's tremendous heterogeneity. And this is in part due to what we call the cancer stem cell hypothesis. So the question is, how and why do we start to see these different populations in cancer in our studies? And the idea is that there are obviously normal stem cells. We have stem cells in every tissue of our body. And these cells give rise to more differentiated cells and can be involved in repair and regeneration, or if they're in the blood, they're going to give rise to your normal blood components. But these stem cells have a unique phenotype. If you had to define a stem cell, it has really two characteristics. One, they tend to be relatively quiescent, which means they don't proliferate a lot. They're your reserve. The other thing is they're capable of either differentiating or giving rise to more stem cells, what we call self-renewal. Now, it's probably a big surprise because the whole definition of cancer is it keeps growing. But what we're finding is in the many cancers, human cancers, both primary tumors and in the cell lines, it looks like that there's subpopulations, and these subpopulations are different than the rest of the tumor. They don't grow as fast, but that means they're also not targeted. A lot of the drugs that we give and radiation attack cells that are proliferating really fast. When you look about chemotherapy, the whole point of a lot of these drugs is you want to literally poison the cells that are growing fast. And when you think of the side effects of chemotherapy in your gastrointestinal and in your hematopoietic systems, it's because those cells are growing fast too, and that's your collateral damage. The cancer actually also looks like it has the same kind of population that may be quiescent. The problem is with chemotherapy, you can only give so much before you can actually, unfortunately, you may cure the cancer, but you're also going to die from the chemotherapeutics. And that's the big paradox, is how much to give for that. The whole property of a stem cell is exactly what I was saying as far as it can give rise to differentiate, but then it is capable of self-renewal. And that's, in that sense, this is a normal stem cell population. A cancer stem cell population is believed to be similar in the sense of it is an escape mechanism for the cancer cell. If you have a cancer cell that's not proliferating fast, it's not going to be sensitive to the chemotherapy or radiation. In fact, there are some cancers, indolent, what we call slow-growing cancers, where they grow so slow, there's nothing they can do. They basically say it's going to take you about 10 to 15 years before you die of the cancer, but there's no way to target it because it grows so slow, so you might as well just go home. And that's the problem, is when you're dealing with these cycle-acting drugs or radiation, it's based on the premise that the cancer cells are growing faster and you can target it. But this is maybe where the immune system can come in, because the immune system doesn't care how much the cells are proliferating. It's just going to attack it as different. And so that's where immunotherapy is coming into the limelight. So the concept of a cancer stem cell is you have in any tumor a small population, and this small population can be only 1%, 0.1%, maybe 10%. And it may be contingent on the environment. If it's a hypoxic area, you may see more of these cancer stem cells. But the bulk of the tumor cells are not that. So what does that mean if you're actually fighting an enemy? This is your smoke screen. This is the one that you're attacking most of the time with a lot of your drugs, and it's going to give you basically what we call a response, or you go into remission. but this population is resistant and waiting to come back. So the idea is maybe this is what we want to ignore almost, sort of like cutting the head off a dandelion, but the root's still there, and we got to get at the root. Because otherwise, obviously, with chemotherapy, these guys are exposed to it too. At any point, mutations can occur that will make you resistant to it. What we're finding too, though, is that there is some evidence that this is plastic, which is even trickier. In other words, you don't have just necessarily this population and that's all it does. We have evidence now that even these cells can revert and they can go back and forth. This plastic thing means that the cancer can evade itself at any point or adapt if we're going to give a stressor to it like chemotherapy or radiation or even the immune system. So we have what I would consider the most formidable foe with regard to trying to attack it, because not only is it mutating and heterogeneous and occurring over long periods of time, but because of this cancer stem cell hypothesis, you might be able to go back and forth and adapt to whatever we try to do at this, as far as treatment. But that being said, though, this does tell us that maybe if we attack it in a 1, 2, 3 punch, just like we did with HIV, we might be able to control it. And control is the key. You're seeing now with cancer, sometimes not the word cure, but control. There have been cases, actually the famous case that I talk about, where this person, a young man, had metastatic melanoma and was treated and was cured. 10 years later, he dies in a motorcycle accident, and they donate his organs. And when you get an organ transplant, you're immunosuppressed. And the people that received his organs actually developed his melanoma. And so what that meant was his body was actually controlling and keeping the cancer at bay the whole time. But from a imaging and from an oncologist standpoint, he was cured. But he wasn't really cured. His immune system was controlling it. And so that's where the immune system may actually give us our out here. So the whole concept of the cancer stem cell hypothesis is that you have the small populations of cancer stem cells in any tumor. And then after cytotoxic therapy, you've basically enriched for this. Now, what that means is these cells are going to give rise to the relapse, and you're going to now have the tumor come back. So we have to target these as best we can. So our treatment paradigm is actually not that complicated. If we know we have a bulk tumor and we have cancer stem cells, if we do standard therapy, we're going to actually enrich for these. But the idea would be then, if we can target this, we're going to have total eradication. So if we do something to target the cancer stem cells, in this case immunotherapy, and either follow it or come first with the standard therapies, this might be the 1-2 punch that we need. So when we talk about cancer stem cells, you're dealing with something that is still a theory. And that's important. A theory being, as opposed to global warming as a theory, this theory is the kind of thing where it's hard to prove. How do you prove that these cells actually play a role clinically until you actually do the clinical trials? And so it's been very difficult at this stage to actually show and prove that these exist in the tumor, in a primary human tumor, other than what we do in our mouse models, where we can actually transplant these cells. And we find that if we transplant based on certain markers, the cancer stem cells, they engraft better in a mouse. They grow better in culture. They're more resistant to the therapies. So we're actually, it's like preparing a legal case where we're actually getting all the circumstantial evidence and all the evidence that we have saying, yes, this is a target. If we do this, we might be able to actually have long-term effects. Every tumor, like I mentioned before, is different. But one thing that we find is these are different cancer stem cell markers on the tumors, and it may not be an absolute. In other words, remember I said that the cells can be plastic, and so it's not like this is a cancer stem cell and this is not a cancer stem cell. There may be what we call shades of gray, and there may be sort of upregulation, downregulation. Unfortunately, people want that magic bullet in cancer. They want the magic bullet that the immune system can see and not see anything else. Now, I've been dealing with immunotherapy for many, many years. I didn't realize it was over 30. Now I do. Thank you very much. But the bottom line is, even when we do immunotherapy, there's toxicities. We're almost inducing an autoreactivity. So the people, for example, that respond in melanoma treatment, they get vitiligo a lot of times. And so what we're seeing is literally some places make a list of organs that you can live without, and if you get autoimmune attack, that's something that's still a viable option versus stage 4 metastatic disease. Okay? But what we see is numerous markers that we have at our disposal. But what we use in my laboratory is aldofluor, because it seems to be, for the most part, common in a lot of the solid organ cancers that we've been looking at. And so we use aldofluor as our readout to tag the cancer stem cell, to measure how many are there, are we seeing effects on treatment. And we use flow cytometry as our readout. This is an aldofluor assay, and what we can see here is this is your cancer stem cell population right here. 5.8% in this tumor line that we have. And more importantly, when we grow out these cells versus these cells, the aldofluor brights grow to a much greater extent. So the smallest population of the cells actually give us our greatest growth potential when we put it in long-term assays, telling us that this is a population that we want to target. One thing that we found, remember what I mentioned before, how do we attack cancer now? We attack cancer with cycle-acting drugs, chemotherapy, radiation therapy. If that's true, things like radiation and chemotherapy should enrich for the cancer stem cell population because they're killing off what we call the 90% of non-cancer stem cells. And sure enough, when we look at radiation treatment, we see the percentage of our cancer stem cell populations go up. And what this is telling us, and this is in vitro, and these are putting human tumors into mice, irradiating the human tumor, and we still see the cancer stem cell population increase as a result. So our current cancer treatments are actually enriching for that cancer stem cell. That's the way the cancer is evading. Now, the cancer stem cell grows slower, so that's a win as far as you're going to see a response. But what we're finding with some cancer treatments, believe it or not, is we see what we call a CRP, or a partial response, or even a complete response. But we don't see changes in overall survival because when the cancer comes back, it comes back twice as fast. Okay? And so what we're finding is, yes, we may be getting a window of time here, but it's only a matter of time before the cancer comes back and is going to be resistant to that treatment that you just gave. So this is a t-shirt that we made in our laboratory when I was at UNR, and then I found out that students would take my cancer class and they'd wear it and they'd cheat off of each other by looking at the back. When I say describe the cancer attack systems, oh boy, that looks very familiar. This is what we call Cancer Immunology 101. The immune system is broken up into pretty much two components, humoral and cell-mediated. The cell-mediated immunity is for viruses and cancer. That's your T cells. The humoral for antibodies is pretty much your bacteria, all right? So if anything, AIDS taught us the importance of CD4 T cells. And CD4 T cells are the orchestrators as far as being able to regulate everything. Now, again, I fell into the trap. I'm drawing all these tumor cells. They look the same. You target one with those little triangles, you've cured cancer. So we've cured cancer many times on pieces of paper, but we now know that the cancer cells is going to mutate. It's not all or nothing. A lot of times the cancer cell is not expressing anything different. It's just more of or less of than what the normal body's making. So Herceptin, which is actually used in breast cancer, that's to a normal protein on the EGFR family. It's just that the cancer cell is more susceptible to block, but you're going to get collateral damage. But this is cancer immunology in a very simplistic form. We got to get these cells involved. Natural killer cells I've been involved with for over 30 years, and natural killer cells are cells that have been characterized by their inherent ability to just what we call naturally kill. Normally, when you get a flu vaccine and you get hit with flu, your T cells come into play, but it takes time. That's why we vaccinate. NK cells are your frontline innate. When you think of the immune system, you're thinking adaptive and innate. Adaptive is your specific arm, your B cells, antibodies, and T cells. Your innate, that's your alarm system. So when you first get hit with the virus or the tumor cells, the NK cells come into play. And they're a fascinating cell type, more complicated than we thought. So NK cells are in your blood. Everybody's got them. In fact, I would be surprised if NK cell discovery, which was in 1975, becomes one of the Nobel Prizes. But they kill in virally infected transformed cells. And they are very attractive for immunotherapy. The problem is the NK cells are in the blood. And so the thing is, if you've got breast cancer, colon cancer, lung cancer, NK cells are not necessarily going to get there. NK cells are believed to play more a role in hematologic, lymphoma, leukemia, but also maybe in metastatic disease, because that's where the cancer cells spread, is in the blood. And so we're thinking of using the NK cells as far as for that therapy. But we developed this hypothesis because NK cells also have this ability to kill stem cells. which is kind of interesting. And fortuitously, we said, well, wait a minute, if there's such a thing as cancer stem cells, maybe we can use NK cells to target that. And NK cells have been used clinically in solid tumors with glioblastomas and everything, and it hasn't worked. And now we're thinking we know why, because we're actually using the NK cells to do it all. And in reality, it's that 1 to 10% of cells that we want the NK cells to attack, not the 99% of the other cell types, which are basically the smokescreen. So when we talk about NK cells, we talk about a cell that plays a role in tumor cell and viral infected cell killing, but it's also regulated. The immune system is not a static environment where it's just one cell affects only one thing. It's regulated by a network of cells, and we have to understand that to understand also how it's inhibited, because that's how cancer is going to evade it, but then also how to exploit it. And we're going to be talking about using some of these cytokines in treatment. And ironically, a lot of these cytokines have been put in people before, and they haven't worked as well, or they cause a lot of toxicities. But now we're figuring out if we do cell therapy, we might actually get a better window because the cell is going to seek out and destroy. And that's the idea with NK cells. So I love these scanning shots. This is a breast cancer cell, and here's our lowly NK cell right here. And the NK cell has numerous recognition mechanisms, and one of them is simply that the cell is not looking right. NK cells have a very crude kind of recognition. It's kind of neat, though, because one of the discoverers of NK cells was from Sweden. And he said, when I was growing up in World War II, when Sweden was surrounded by enemies, the Navy was having a hard time figuring out friend from foe. And so basically the Swedish Navy said, well, we're small. This is what we look like. Kill everything else. And that's sort of like what NK cells are like. NK cells, this is what I look like, and if it doesn't look exactly like this, I'm going to attack it. All right? And so we're using that to our advantage. Now the NK cell is actually binding the breast cancer cell. Now it looks like a piranha feast right here. Here's some NK cells all on this big breast cancer cell. And the way NK cells kill is actually beautiful. They poke a huge hole into the cell. And what that does is it disrupts the cell membrane, and the cell basically blows up and spits out its guts. And then the NK cell moves on to another cell, and what we have is a skeleton of the cell. All right, so it looks great as far as when you do this. It's harder to do it in people, all right, because you've got to get the NK cell to every cell, all right, and they have to be able to see it every time, and that's been the big problem. So what we do in our laboratories, we do human NK studies. And so what we do is we first have to get NK cells from normal human volunteers. There's a sign-up sheet at the end of the hall here. Yeah, people got nervous. But then we isolate these cells through a variety of procedures, and we grow them up. Once we grow them up, we activate them. And these are activated killers. And so the idea would be then we're not going to use these as an end-all be-all. We want to enrich first for the cancer stem cell and see if the NK cells can kill it. And the way we do NK killing assays is not that complicated. One is we just label cells, tumor cells, with a radioisotope. And remember, the NK cells poke a hole in the cell. All that radioisotope gets leaked in the supernate, and we can measure that as a % killing. Or we can do a flow cytometric assay, which we actually look at the type of killing that can go on. But these are our readouts in our assays. And one thing that we found, which is really interesting, is these are different human tumors. These are all human: breast cancer, glioblastoma, Ewing sarcoma. We see that small population, sometimes not so small, but sometimes it's in single digits. And if we mix the cells together with the NK cells, we see it goes from 28% to 2%, 9% to 3%, 7% to home run, 0%. The NK cells are preferentially killing the cancer stem cell. And this is in a mixed population, so that's telling us we can actually get this even when there's a lot of smoke screens around. But the question is, if we reduce this first, now if we can get the NK cells to actually mediate the effector function. So when we look at this too, it's important to keep in mind, one of the advantages of UC Davis and being at a cancer center and a medical center is I actually have in my laboratory a surgical oncologist, a radiation oncologist, a hemonc. And we can get primary human tumors. That means first thing you want to do if you have cancer, usually, is cut it out. Cut out the primary tumor. Well, we can actually use that to validate what we're seeing as far as with all those cell lines. And we actually incubate the NK cells with the primary tumors, and do we see the same effect? So we do the surgery, we excise the tumor, we process it, we do an NK cell from a normal donor, and then we co-culture and we look at the effects on cancer stem cells. And what we found, to our pleasure, is that we see a similar pattern, inasmuch as these are primary human tumors, literally taken hours from the patient. Because tumors, again, people don't realize this too, is that the tumor cell lines that we use them for a reason. It's actually, most human tumors, when you put them in culture, die. It's really hard to grow up a human tumor line from a primary tumor. And so what we're finding here is in 24 hours, we see again the reduction of cancer stem cell markers in the primary tumor, suggesting that we might be able to apply this in human cancer clinical conditions. However, all the in vitro assays are great, but ultimately, we're going to have to figure out, does this work in vivo? Why? Because ultimately, we got to get the NK cells to the site. All right, it's got to target it. And so what we've been doing is a variety of tumor models. Some of the tumor models are actually kind of fancy, and I'll go into it, but some of these are using special immunodeficient mice. Normal mice will not allow human cells to engraft. This is not a normal occurrence in nature. And so what we have to do is use special mice that allow these human tumors to engraft and these human NK cells. And basically, we're using the mouse as a test tube, but a test tube that adds a little bit more complexity because you have the normal organs, normal trafficking, toxicities, everything else. You can't just now, for a human trial, say, this looks good on paper, I got the in vitro data, roll up your sleeve. You can't do that. You need some sort of efficacy studies, and that's what we've been doing here. And what we found is when we do primary, and well, not primary, but these are just human tumors, and this is a pancreatic cancer, by the way. What we find is when we co-inject NK cells with the tumors, we see a lot less of the aldefluorobright positive population. We're targeting the cancer stem cell. And that means we're starting to see effects that maybe we can exploit when we combine it with other therapies. More importantly, if we actually start looking, CD56 is an NK cell marker. So when we actually take these tumors out, section them, and we start to look on where the NK cells are and where the cancer stem cells are, this is an NK cell marker, this is a cancer stem cell marker in green, and then we start to see the NK cells going to where the cancer stem cells are. So the NKs are actually seeking out the cancer stem cells and trying to find it. And sure enough, we see a reduction when we look later on, 5 days later, for the presence of these cancer stem cells. So the NK cells seem to be working. The question is now, can we tie it all together? What's also important too is it's important to look at a clinical situation like lung metastasis. When you talk about metastasis, you're talking about areas where there's usually high blood flow, the lung, the liver, the brain. This is what makes cancer so difficult as far as these are vital organs, but also the therapy has to get there. And what we found is when we inject these mice with the human breast cancer cells intravenously, the tumors will develop in the lung and the liver like a metastatic tumor. And more importantly, when we give the NK cells, we find they go to the mouse lung and they seek out the human tumor, and more importantly, the human cancer stem cells, and attack it. So this is a good thing as far as proof in concept, that at least the cells are meeting up, all right, and that's a good thing. More importantly, when we actually then take out the tumors, as far as we take out the mouse lungs that have the human tumors and we grow out, seeing what grows out as far as tumors, when we give tumors versus tumors and NK cells, we get a lot less human colonies forming, and look at the difference between the size of the colonies. In other words, this is the tumor cells that we get from the mouse lungs, the human breast cancer. When we add the NK cells, they're still there, but there's a lot less of them, which is a good thing. Okay, can we improve the recognition? We got to gear the odds in our favor, because the NK cells, already the cancer cell is going to try to escape. It's going to try to evade. And so one thing that we found is when we give some of our conventional cancer therapies, like radiation and some chemotherapies, it actually not only kills the non-cancer stem cells, but it upregulates death receptors on the cancer stem cell, making it more attractive for the NK cell. So it's sort of like getting more bang for your buck. It's just a matter of now we had to put it together. And these are basically different kinds of NK ligands or death receptors, and the dotted line is after radiation. We can see it goes up, which is a good thing. And they also show the cancer stem cells seem to preferentially show an upregulation of these. The non-cancer stem cells, they die You give chemotherapy, radiation, they're proliferating, they're going to die. The cancer stem cells seem to upregulate these stress ligands, and that's making the NK cell very happy because that's increasing the bullseye that we want to use. So one thing that we wanted to do, a proof in concept, is we have the tumor lines, we irradiate the tumors, then we mix them with the NK cells, and now we want to look at the total cancer stem cell frequency and just overall killing. If we just look at live tumor counts, this is without NK cells and with NK cells. This is basically your tumor cells growing. Radiation, well, we knew radiation would affect tumor growth, and we're seeing a reduction here. When we just give NK cells, we see a reduction, but when we give the combination of radiation with NK cells, we get our greatest kill percentage of our tumor cells. And this is important too because we're also seeing increased cancer stem cell killings. So what this is telling us is we may be able to use this to our advantage, but now we got to see if it works clinically. The other thing that we're looking at is not just radiation, but Bortezomib, molecular targeting agents. You hear that a lot, molecular targeting agents and targeted approaches in cancer. What we're looking at is ways that we can exploit the cancer cell proliferation to our advantage. It's basically not that much different than a chemotherapeutic per se, but it's actually something that has a little less toxicity, so it's very attractive. We've been interested in Bortezomib, which is a proteasome inhibitor, for some time. It's actually currently used in lymphoma, but we're using it now, and it's actually been tried at UC Davis for lung cancer. It didn't work. Well, now we think we know why it didn't work, because we can use it for the cancer stem cell targeting, but just not the rest of the tumor. And so what we first did is we show that also when we give this molecular targeting agent, Bortezomib, co-incubating with the tumor line and then the NK cells, we look for a killing. And we again find that Bortezomib actually sensitizes. Again, when we're looking at Bortezomib, this is the aldophluor bright population. This is your cancer stem cell population. It drops down to 0. And so what we're finding is that we can apply other agents with immunotherapy to target the cancer stem cell. So now the proof in the pudding comes to survival models. And this is not easy to do because ultimately, too, it's very easy to cure cancer in mice. The reason being is when you're giving cancer to a mouse, you can wait 3 days. I've seen some people where they treat the animal first, then they give the cancer, and surprisingly, the cancer doesn't grow. All right, so that's as artifactual as you're going to get, versus in a human situation where you've had cancer over long periods of time. All right, and it's very heterogeneous. So what we want to do is we want to use the mouse as a model, but we have to make the models more realistic. And that means we want to use them in the sense of how we would treat a real patient. So one model that we're using is glioblastoma. Now, glioblastoma is one of those rare cancers which it will kill you not because it's metastasizing, but literally because it's an area in the brain that you can't get to, and it just keeps growing. So it's one of those cancers that will spread out in the brain, but becomes inoperable. And then once it becomes radiation resistant, there's really not much, or chemotherapeutic resistant, there's not much you can do. And it's got a very, very poor prognosis. And so here's a glioblastoma in a patient. These people have very little options, but it's in the brain. And so the question is, how can we use our treatment regimen to actually treat these patients? And so what we do is we use what we call a stereotactic model. And this is actually a lot harder than it looks because I can't do this. But this is where we actually will inject the tumor into the brain to mimic a human glioblastoma. This is a human glioblastoma into a mouse brain. But then we want to give the therapy in the brain. So the idea is we're going to treat with either chemotherapy, radiation, and you can do that locally as far as with brain radiation, but then you come in with your NK cells. So this is what we call an orthotopic model. Rather than just inject the tumor into the flank of a mouse, we're injecting it into the organ where it normally grows in, making it more realistic. So we're giving the tumor intracranially, then we come in with NK cells in the brain. And then what we're going to do too, in some of these studies, we're going to be also giving either radiation or Bortezomib. One nice tool that we have is by using a glioblastoma cell line, we can use a luciferase tag. So we can actually image it. In other words, we can actually see the tumor in the mouse as it grows by bioluminescent imaging. And you can see with the increased signal and intensity the growth of this tumor in the mouse brain. This is really nice because it gives us, instead of looking at simple survival, it's telling us how the tumor is progressing and then also how we can measure efficacy as far as literally by measuring the intensity. And more importantly, we don't have to euthanize an animal each time. We can image it, the same animal, over periods of time, which is really nice. And so what we find here is putting it all together, doing the imaging. We have untreated mice, mice that had NK cells, mice that received Bortezomib, and mice that received the combination. The combination is that 1-2 punch we were hoping to get. This is the growth unfettered of a tumor where these mice eventually will die pretty rapidly because of massive spread. What we see is, even at these cases, the NK cells are providing a protection. But much like the clinical trials, it's not going to work by itself. Bortezomib alone has a very modest effect. But this is where when we gave together, and this is just one injection of the NK cells. Obviously, in a clinical trial, you're going to be doing multiple injections of a therapy. Just one injection, we're actually seeing a significant reduction in tumor burden and increase in survival. And these are actually scans of the mice also showing the treated versus the untreated as far as the tumor presence. So what this is telling us is by targeting the cancer stem cells by using a drug, either Bortezomib, and I'm going to show radiation in a little bit, then coming in with the NK cells, we might be able to see greater anti-tumor efficacy. One of the worst tumors there are is pancreatic cancer. And pancreatic cancer is one of those rare cancers, too, where whether you're stage 1 or stage 4 being metastatic spread, stage 1 is being the lower tumor burden, very contained, the mortality rates are actually pretty comparable. It's very difficult to treat. It's very aggressive and very hard to actually study. In this study, we actually injected the human pancreatic cancers, waited for the tumor to develop. Now we're giving local radiation, and then we're injecting the NK cells. And what we find here, here's your untreated mice in black, which you can't even see. This is NK cells in blue, so it's superimposable. The NK cells had no effect on the pancreatic cancer, which is probably not surprising. When we give radiation, well, radiation is your frontline treatment for many cancers. We do see a protective effect of radiation. Unfortunately, the tumor does come back, and not only that, but if we irradiated the tumor again, it would have no effect, so it's resistant to radiation. Actually, Eric, that's a good experiment. We should do that. I'm sorry, my students are here, and I just thought of that. See, these talks actually help me, too. But when we give the combination of NK cells with the radiotherapy, we're seeing a significant reduction of the tumor growth, and this is evidenced also by survival, significant increases in survival. The mice die within 75 days, whether you give untreated or NK cells. This would actually be enough almost to do a clinical trial in pancreatic cancer. I think one of the drugs increased life expectancy by about a week, and it got FDA approved, just because there's nothing out there. But when we give radiation, we do see an effect, but that more importantly, it's the combination that we actually see our greatest protection. So this is telling us that what we saw in vitro, our model, has merit, and we can actually see increases in survival. Where are we going with this? Well, most people don't realize UC Davis has probably one of the best vet cancer hospitals in the world. It's actually, at one point, they had a bigger dog cancer hospital than a human cancer hospital at one point, as far as Davis Cancer Center versus the Vet Cancer Center. They treat over 450 dog cancer cases a year. Now, why do we want to use dog? Because using inbred mice is nice. I know that rhymes, but mice is nice, but it's not the same as a large outbred population with spontaneous tumors, and that's the advantage of the dog model. The other thing that we can address with the dog that you really can't deal with mice is what we call quality of life indexes. As far as people that have cancer or have to undergo cancer have to deal with at some point, your quality of life as far as the amount of chemo radiation that you get is going to impact as far as what you go for and how long you pursue it. And pet owners are extremely sensitive to those kinds of issues when they take their pets in for treatment. Now, dogs don't get people cancers as far as in the classical sense. They get sarcomas, they get lymphomas, and they get melanoma. primarily. That's what we get at Davis. So what we want to do now, we've already started the preliminary studies, is we're going to actually take dog cells, peripheral blood, and grow up their NK cells. We already can do that. But what we're going to do now is treat the dogs with local radiation, and then we're going to come in with NK cell infusion for proof and concept, both looking for efficacy, but as also quality of life indexes and as well as toxicities. What's really nice about UC Davis is they have cutting-edge imagery such that we can do PET CTs on dogs, and we can do MRIs on dogs to actually visualize the tumor. So the idea is this is sarcoma that's already spread to the dog's lung, and we're going to try to treat either the primary tumor, which the dog presents with, or we're going to give systemic NKs. We're going to try both populations. This is a dog sarcoma, and we've already done studies in dogs looking at radioimmunotherapy. That is, we give radiation treatment, and then we give local immunotherapy, in this case using CPGs. And these are a scan of a regression of a primary tumor in the dog after treatment using this. And instead of CPGs, now what we're going to do is we're going to use NK cells as our therapeutic regimen. And so these are where we're going as a prelude to the clinical trials that we want to go forth on. So what can we conclude from this? Well, it looks like the cancer stem cell is a very attractive target, but it also is a very elusive target because it's resistant to many things that we give it, and if there's plasticity involved, it's going to be a constantly moving target. So the idea would be then we're going to apply cytotoxic therapy and NK therapy. What we don't know is the timing, because obviously we want to make sure we get maximum effects of both. And at the same point, many cytotoxic agents, guess what? They suppress the immune system. And so it actually can hurt us if we apply too many cytotoxic regimens or chemotherapeutic regimens. It's going to kill the immunotherapy that we're giving it. In fact, that's one of the paradoxes in cancer is the fact that many of the people that we treat with cancer are over the age of 55. Their immune system is already hampered because they've been getting massive chemotherapies and radiation therapies, and yet we want the immune system to mop up the rest. So the idea would we would leukapheresis first the individual, get their NK cells, then give the chemoradiation, come in with then the NK cell therapy. I think we've shown that NK cells can actually not only target the cancer stem cells, but actually preferentially target them. And when we use radiation and bortezomib, we can actually sensitize the cancer stem cells to NK killing. And this combination, radioimmunotherapy or chemoimmunotherapy, may give you the best chance, not for remission per se, but for long-lasting control. And our future goals are a canine trial, which we're actually initiating now, but ultimately we want to do a clinical trial. I'd like to acknowledge the people in my laboratory. Eric, right here, he's back there. He's the one that actually did the talk. He got this all ready for me and has actually done most of the studies, and I don't know why he's smiling. He should be at work. But the bottom line is Bob Cantor, Dr. Cantor, is a surgical oncologist that's helping us with the sarcoma studies and pancreatic cancer. Dr. Arta Monjazab is a radiation oncologist who was pivotal in the dog radiotherapy as well as doing, starting the clinical trials. Numerous students. Where's my CERM students? Do I see them? There's one. There's Stephanie. RJ, guess, were you in there, RJ? No, you were late again. Okay. Stephanie's there. She's a CERN Bridges student, and Janelle is a Bridges student from Humboldt. A fantastic group of people, and these are all the different collaborators at both NCI and Michael Ken at the vet school. Thank you very much.
So I used to have a neighbor. She's still living right now, but she's a heavy smoker, and I've heard that some people have a cancer gene. I'm sorry, smoke gene. Is that true?
Have what?
Smoke gene.
Smoke what?
Gene.
Smoke gene. What's the smoke gene? Do you want to smoke?
But I mean,
no. You mean as far as susceptibility to cancer because of the...
Right. She's a heavy smoker, and I'm just surprised that she's still alive.
Oh, I see.
65. She's like 65 years old.
Yeah. So you know, clearly we always hear of those cases where somebody's done everything wrong and don't have cancer. You know, what's fascinating about, lung cancer in particular, if you look at a pathology textbook from the 1800s, they don't even talk about lung cancer. In fact, one textbook apologizes for bringing up lung cancer because they say you'll never see that. And it was so rare in just medical practices. Now, lung cancer is relatively common, but more importantly, we're seeing an emergence of non-smoking lung cancers, which was also unheard of. And so, you know, clearly there are going to be genes as far as DNA repair, like, for example, BRCA1, BRCA2 that you hear for breast cancer and ovarian cancer, that makes one susceptible. There may be also genes that makes one more resistant. That doesn't mean that you want to play the Russian roulette with an automatic by doing a lot of smoking to say, well, I may be resistant. So I think that there may be a case, clearly though there's overwhelming evidence, the decrease in lung cancer mortality is actually coinciding with the smoking cessation in the US. So, you know, we're clearly seeing a benefit by not smoking. But I think that there may be resistance genes and maybe immune system and maybe repair. But I don't think that's been well worked out. People are looking more at the other end of the spectrum for susceptibility genes. That's the thing, and that's where you see it in families and things like that. And that's actually probably more indicating that something's going to happen regardless of what you do or you want to limit exposure. So if that helps.
So for the IVIS studies with the mice, I was wondering what was the transduction efficiency of the cancer cells?
As far as with the luciferase?
Yes.
So what we've done is these are lines, established lines that we've grown up and maintained over long periods of time. And so we don't worry about them kicking it out. For the most part, every cell's been selected so it always expresses it, and it's a stable transfectant. Yeah.
You were mentioning about when you take primary tumor cells and you put them in culture. You mentioned under normal conditions, they're already difficult to develop into cell lines.
Yeah.
I'm wondering, once you started cold culturing NK cells with primary tumor cells, how did you detect that it was the NK cells and not the...
That's an excellent question. So you walk this fine line between when cells start dying after 24, 48 hours. So what we do is we look earlier than that, at which point we compare it to the cells that don't get the NK cells. And if we see the differential, it's not optimal, but it's still better. Because the other thing is, I don't really mention it, is these cell lines that we've cultured are kind of artifactual. Normal human cancers have a lot of normal cells. In fact, some cancers have more normal cells they've recruited and pirated than the actual tumor. And we can't mimic that by using the cell lines. The primary samples that we have are mixed with everything. And so that's more of a reflective of what the NK cells can actually come up against. Because they also have to see the cells, the cancer's actually pirated normal cells to suppress the immune system. So sometimes it's not even getting the immune cells there. It's once they're there, they're turned off. And so we're trying to look at ways to get around that as well. But for the most part, by looking at that window short term, which limits, we can still make that differential that we're seeing in effect.
Have you tried to look at NK cell survival since you're injecting them into like tumors and stuff
Yeah,
like that?
so we've done both injecting them systemically as well as in the tumor. And that's an excellent question. And the tightrope is, when we grow up our NK cells, we activate them. We give cytokines and they're cranked. But that means they crap out. That means after about 1 week, we don't see them anymore. That's all they're going to do. There's a fine line between, in cancer therapy, control versus impact. And so what we're trying to figure out now is if we don't activate them as much, do they live longer and therefore have more of an effect? Or does that give also the tumor a chance to evade? And so we're trying to figure that as far as that fine line between going for the knockout punch versus going for a lot of low body shots such that we're controlling the tumor. And that's because the other thing too is we're using NK cells in a mouse. We're hoping, and we're doing these studies now, that also your other immune cells will say, hey, this is working, and start expanding and attacking too. Because, you know, that young man with melanoma probably wasn't his NK cells, it was probably his T cells, his other immune cells that were keeping that tumor at bay. And so what we're hoping to do is get the NK cells to get the ball rolling, but then the immune system will come in and finish it up.
You said the NK cells can attack the cancer stem cells.
Mm-hmm.
Is there a way to use memory T cells also to activate NK cells to use?
Yeah, and in fact, there's a lot of work looking at that. One of the things that, you know, there's always a positive spin. One of the positive spin on AIDS and HIV is we learned on how the CD4 T cells affected all the other cell types. And so what we're finding out is if we actually get the T cells involved as well, we might have a longer lasting effect. And so we're starting to look at some of those components as far as the T cell component versus the NK. But you're exactly right, they feed into each other. But it's just going to be a matter of how we're going to control it. So the immune system is also really geared for being turned off, because autoimmunity is nasty. And I didn't really show some of, even though we're getting some home runs in immunotherapy, some of the toxicities are really tough because it's like putting gasoline on a fire. We can't stop it. The advantage of NK cells is that what we call terminally differentiated. So normally when you do a cell therapy, you have to put a suicide gene in such that you can stop the therapy. Otherwise, you can't control it. With NK cells, we know that they'll eventually die. And so it's actually an element of control where we can hit them multiple times, but we know we don't have to worry about long-term collateral damage. T cells, we might have to worry about that.
So I'm kind of curious that if you guys do get it to where the body can suppress it itself, say like with the young man with the, we have our natural cells killing it off. Are you worried about like long-term effects as far as possibly affecting maybe like organ donations and stuff like that?
Well, organ donation is probably the least of the patient's worries, but what I'm worried about is some of these therapies literally don't stop. So some of the T cell therapies, for example, to a lymphoma, B cell lymphoma, they keep going, and then, remember I said you can make a list of organs you can live without? Well, B cells are one of them because your body makes more. The problem with some of these landmark therapies is you're B cell deprived for the rest of your life, and they can't stop it. And so toxicities are always a major concern. When I was at the National Cancer Institute, we called it the National Cytokine Institute because we just would dump IL-2 into people and things like that. And IL-2, there's a 2.2% mortality rate that it will kill you. So if it doesn't cure you, kill you kind of a thing. 16% survival as far as from the cancer, but 2.2% death rate just from the treatment. And so that is a huge concern with the NK cell therapies that we're not going to see in a mouse model because the human NK cells may not see the mouse tissues as something that they're going to want to go to. The dog studies may tell us that.
When you introduce NK to a region where it doesn't have that in that area, is the immune system attacking it and therefore you're using immune suppressant to prevent it from attacking the NK cell?
So there's two ways to use the NK cells. One is you use what we call autologous. That means we take your NK cells and put them back into use. So your body's going to be fine with that. But where a lot of big places like MD Anderson and Sloan Kettering, they're going radical. They're doing allogeneic NK cells, where they're going to take NK cells from another donor, immunosuppress you, almost give you a bone marrow transplant, and then come in with the NK cells because it's been shown that they kill tumors better. But then they have to worry about your system's going to reject it. And so that is the fine line between great anti-tumor effects, but more toxicities or chances of rejection. So for the most part, with an autologous scenario, you don't have to worry about that.
I think that ran the gamut here of the questions. Let's thank Dr. Murphy once again.
Thank you very much.